Okay, good afternoon, everyone. And from now on, the rest of the semester, we're going to be discussing bacterial disease. I might have one or two references to non-disease causing bacteria as well, but our primary focus is now going to be on clinical bacteriology. And we're going to begin with one of the really important sets of diseases, and these are diseases of the respiratory system, still responsible for an enormous mortality of the human population worldwide, as we'll see. You excuse me because I actually have a cold at the moment, so if my voice starts going a little bit wonky, you understand why I have a respiratory disease of the upper respiratory tract. So, okay, so let's have a look. I want you please to go back and revise your what you studied in A&P about the anatomy of the respiratory system. And I'm only going to briefly refer here to protective mechanisms that are present in the respiratory system, remembering that there are different kinds of chambers of the respiratory system, which are quite separate from one another and yet connected. First of all, the nasal cavity and the sinuses. The nasal cavity and the sinuses have a rich microflora, which is a protective mechanism because the microflora are natural, they are tolerated, and they actually exclude pathogens to a large degree. But to stop overgrowth of those, etc., because of course many of them are potential opportunistic pathogens. We have a rich production of mucus in the cavities, and in the case of the nasal cavity, that mucus traps bacteria, which are then either blown out when you blow your nose, or else swallowed. The vast majority of the mucus which we produce, which is produced specifically to trap and deal with bacteria and small fine particles, the vast majority of that is actually swallowed. In the mouth, there is an immense bacterial population. And again, we swallow continuously, and we kind of trim off the bacterial population continuously so that it doesn't overgrow. And that in itself is protective. The bacteria growing here, which are completely natural, exclude pathogens. And I also want to point out there is a danger here. With such a rich bacterial population here in the mouth and the nasal cavities, we are continuously producing aerosols which have the potential to be inhaled through the trachea and end up in the lungs or in the lower respiratory tract. So there we will discuss the protective mechanisms that exist in the lower respiratory tract in a minute. So the lower respiratory tract consists of, first of all, leading to it is the pharynx or the throat. The pharynx is actually a unique anatomical feature because it is a union between esophagus and respiratory tract. And there is a protective mechanism to prevent food from going down the respiratory tract. And that is this piece of cartilaginous muscular tissue called the epiglottis. The epiglottis, when we swallow, the epiglottis closes to close off the trachea so that you don't inhale food. The lower respiratory tract is initially reinforced with cartilaginous rings. And it's, first of all, here is the voice box, the larynx, and then the rest of these are the large tubes are the bronchi, and the bronchi are richly invested with ciliated epithelium, and that ciliated epithelium in turn is covered with layers of kind of gel, really more than fluid, and the thickest layer is the mucus layer, and the cilia tend to conduct that up the bronchi and then up through to the pharynx where it is swallowed. As the bronchi divide down deep in the lungs, they divide into bronchioles. And the bronchioles are not actually that richly invested with ciliated epithelium. They do have some. And then they subdivide into the alveoli at the bottom here. The alveoli have no mucus covering the surface. Instead, they are covered with a surfactant. But all of this fluid operates to form a sheet, which is conducted up the bronchi and then to the pharynx where it is swallowed. And that is called the bronchociliary escalator. And the bronchociliary escalator is one of our most important protective mechanisms because it traps particles as they move down the bronchi. They get trapped in that mucus, and they get conducted up to the pharynx where they are swallowed. So they are kept away from the lower respiratory tract. The upper respiratory tract is rich in bacteria, most of them our own microbiota, but the lower respiratory tract is practically sterile. This mucus is not just snot. Mucus here is a very, very active substance. It contains protective chemicals. It contains antibody, which also serve to act against bacteria trapped in it. And down here in the alveoli, there are dedicated macrophages called the alveolar macrophages, which continually survey the surface of the alveoli and phagocytose anything that they find, not only bacteria, but particles as well. And in addition, there is a specific antibody produced in the alveoli called immunoglobulin A, which is IgA, which again has protective function. All of this serves to keep the lower respiratory tract practically free from bacteria. You might find a few, but they do not survive to reproduce unless something is going wrong. They are soon conducted out of the lower respiratory tract. So here are the, this would be typical bronchial ciliated epithelium. And here you can see the cilia here. These large cells here are called goblet cells, and they're what produce the mucus. This is a scanning electron micrograph of the cilia. So this bronchociliary escalator is the most important mechanism, and we're going to hear that can sometimes go wrong, and that some pathogens have specific virulence factors that interfere with this. So keep an eye open for those. When things do go wrong, it will most commonly set off an inflammation, and that is usually what we register most as our first symptoms. And the typical thing for most of us, we've all had, is pharyngitis, which is inflammation of the pharynx, and that is your typical sore throat. And that can extend, most commonly, it's right at the top of the pharynx, behind the mouth. But it can extend further down the pharynx, and it can actually become a rather dangerous situation, as we'll hear for some specific diseases. This inflammation can set off a cascade of events which may become life-threatening. Under normal circumstances, it's not. Most of the time, pharyngitis resolves itself. But often, as you know, it's extremely painful, and it's very limiting when you have a very sore throat. The most dangerous situation is, in fact, when the epiglottis becomes inflamed, and that is often as an extension of the pharyngitis. Epiglottitis is inflammation of the epiglottis, and this is a life-threatening situation because it can lead to the closure of the airway. And under those circumstances, this is an emergency situation, and it needs to be dealt with vigorously in order to preserve the life of the person. Laryngitis may also be familiar to you. I'm just having a little bit of a moment. Very, very common when you have a cold. And it's this inflammation of the larynx tissues, of the vocal cords. And again, it's usually just a bit of a nuisance and self-limiting. Now, tonsillitis is a little bit different. The tonsils lie at the top of the pharynx. They're actually part of the gut immune system structures. And they are constructed in such a way that they have deep clefts in them. And they are a very strong protective mechanism which prevent pathogens from entering the gut, especially. But the problem is that they can get overwhelmed, and bacteria can actually settle in these crypts in the tonsils and begin to reproduce, rather. And they produce inflammation, which is intensely painful. And this is usually in response to bacterial infection, can sometimes be due to virus as well. This is very common in children. And in the old days, they used to immediately whip out the tonsils. But that is nowadays, in modern times, that is not commonly done. And most of you probably still have your tonsils. Adults certainly can get tonsillitis, but it's much less common. Sinusitis is infection and inflammation of the sinus mucosa, and it is usually due to an interference with the drainage system. The sinuses drain into the nasal cavity and into the throat, and that drainage should be free, and sometimes even a mild infection or inflammation due to allergies, such as pollen allergies, that sort of thing, can interfere with the drainage and lead to infection and inflammation of the sinus. And as those of you who suffer from sinus know, this can be intensely painful as well. And most of these bacterial infections of the upper respiratory tract are actually self-limiting, but will often take antibiotics to shorten the length of the infection, etc., etc. That may not be that good idea because antibiotics are so commonly overprescribed that it leads to resistance in the populations that are causing the problem. Okay, so let's have a look at some of the specific infections, some of them extremely important and some of them extremely dangerous. These are bacterial diseases of the upper respiratory tract, from, let's say, from the larynx on upwards. So the most common thing is pharyngitis. And the one organism of particular interest in pharyngitis is Streptococcus. And this most commonly Streptococcus pyogenes. Now, the Streptococcus pyogenes is a common skin bacterium. It's one of our commensals. It also does occur in the nasal cavity, but it is kept in control by all of the other bacteria that grow. But if we disturb the population structures of the throat, then especially if you disturb the mucosa, then it predisposes to an infection with a streptococcus. And the most important group are group of streptococci, which belong in, they're classed here in what is called the group A streptococci. And group A streptococci are beta hemolytic streptococci. And if you go back, please, and see what beta hemolytic means, this is a very important distinguishing factor in trying to decide what is causing a pharyngitis. And this is one of the things that will be tested for. So Streptococcus pyogenes has numerous virulence factors. That is, they have things which predispose them, causing inflammation and for being able to reproduce on a mucosal surface, and also internally as well, should they get there. The first is that these group A streptococci are resistant to phagocytosis. Phagocytosis is first line immune response. The macrophages phagocytose the bacterial cells and actually inform the rest of the immune system that something is going on. So it's a primary response to the presence of a pathogen. That's the first thing. The second thing is that Streptococcus resists phagocytosis by neutrophils, and neutrophils are the second line of response, and they are the soldiers of the immune system. And they come in to specifically to a site of infection to phagocytose bacteria. But Streptococcus is able to resist this phagocytosis. The second thing, the virulence factor they have, is they produce a set of enzymes called the streptokinases. And it's a little bit difficult to describe, but you need to think about what a blood clot is constructed of. Blood clot is constructed of strands of fibrin. That is a protein which is laid down when the clot forms and which traps red blood cells inside it. One of the immune responses which can be used in bacterial infection is that the immune system will cause the formation of a blood clot around this area of infection. But the streptococci produce these streptokinases, and they actually break down the fibrin. So I have to say that's an oversimplification of what happens, but in effect, the fibrin is broken down and the clot dissolves and releases the bacteria that it was trying to enclose. And in addition, they produce a set of enzymes called streptolysins, and these actually cause cells to die. And what that enables the bacteria to do is to reproduce in the area and have a rich source of nutrient as well. So all of these things acting together can cause an intense local inflammation, and all of the effects of that, all of the inflammatory responses that we've talked about before, swelling, pain, fever, enlarged lymph nodes, all of these things are actually immune responses, but there is cellular damage that is taking place as well. So this is ordinary kind of streptococcal strep throat. I think I've got a picture. Here we are. So this is typical strep throat with an intense inflammation, the redness here, and these white dots here are not highlights or reflections in the back. These are actually bacterial colonies growing there, and just as they would on an agar plate or something like that. So this does need to be treated if it persists, and it does need to be treated in children very often with antibiotics, because we're going to hear that it can lead to some other very severe illnesses. But the majority of sore throats are caused by other things, other bacteria. And there are just a great number of them that can cause it, and almost all of them are self-limiting. If you just leave them alone, they will go away. If the symptoms like fever and swollen lymph nodes persist, then medical attention does need to be found. The distinction, but you realize it becomes important to distinguish between all of these other things and streptococcus. And so there are a number of tests which can be done, quite apart from just a simple smear that looks under the microscope to see what you've got. There are a number of other tests which can be done, one of them being that you would plate it and see what it's plate it onto blood agar and see what its hemolysis pattern was. The reason strep needs to be kept an eye on is that it is possible for some of strep strains to have been infected with a virus. And this is a specific set of viruses which are lysogenic viruses. That is, they can remain dormant, essentially, inside a cell by integrating into the host genome, and they just get reproduced every time the cell reproduces. But in addition, though, that integrated piece of viral DNA contains virulence genes, can contain virulence genes. And one of the virulence genes, which is really important in Streptococcus, is that it produces a toxin which accumulates inside the bacterial cells, which is then released when the bacterial cells die. Whereas they're growing, any population is going to have a certain death rate. So you've always got cells bursting and lysing and releasing their cell contents. And when this toxin is released into the bloodstream, it can produce a specific set of signs which are erythrogenic. This toxin is erythrogenic. It causes reddening of the skin. And so here, this here is this condition called scarlet fever. And scarlet fever produces this kind of mottling red of sign of the skin, hence its name. And the release of these toxins is usually either because the bacterial cell has just died, or because the virus has entered into a lytic phase and has caused the lysis of the cells. It in children can be dangerous because it can cause a very high fever. It has some other signs. Here, this is called strawberry tongue, and it's this glossy, swollen, scarlet tongue. So that's the first thing. The scarlet fever in itself can, in fact, be dangerous, but it's, again, usually self-limiting. It used to be extremely common in children, and certainly had a certain mortality associated with it. But it's really important because it can be a precursor to rheumatic fever. And the reason that rheumatic fever is so important is it's not actually due directly to anything that the bacterium releases or anything like that. The immune response to these lysogenized bacteria produces a set of antibody against proteins which are very similar to some of our own protein. And it is possible for our own antibody to begin attacking our own cells. And these cells are particularly common in the joints and on the heart valves. And for that reason, this condition will be called rheumatic fever, because there's a swelling of the joints as the immune system attacks our own tissues. And the grave danger of rheumatic fever is that the heart valves become damaged. And in the old days, before vaccines and everything else, this was a common cause of severe heart disease in children, and then, of course, into their adulthood, it didn't repair. The problem with rheumatic fever is subsequent streptococcal infections can actually reinvigorate this autoimmune response. So it's a very severe condition, but fortunately extremely rare, even long before the advent of antibiotics and everything else. So that's one lot. So here's the next one, and this is Corynebacterium diphtheriae. And this is a really severe illness which used to have a very high mortality in children. It used to be one of the commonest causes of death in infants. And in the 19th century, almost every family could count on losing at least one child due to diphtheria. And often entire families of children would be lost. A very, very dangerous disease and quite easily transmitted. and as a result, the cause of real anxiety to families, and one of the reasons why people used to deliberately have very large families, because they could count on losing some of their children due to diphtheria. So this is a gram-positive bacterium, and it is interesting because, in actual fact, this is one of the bacteria which, in and of itself, our immune system can deal with quite easily, as long as it doesn't possess as long as it's not able to produce an active toxoid, a toxin released into the bloodstream. Released into the bloodstream, but also released locally. Its major impact actually is local. And so this is bacterium settles usually, the infection would usually be in the back of the throat. And it releases this toxin, which causes the membranes of the throat to form a thick membrane. It's called a pseudomembrane. And that pseudomembrane is what causes the real problems. And it is that pseudomembrane formation is due to the release of this toxoid. That toxoid, again, is produced by genes introduced by a lysogenic bacterium. And it causes the accumulation of the toxin in the cells, and just as we saw for streptococcus, when the cells lyse, either natural because they just die, or when the virus enters a lytic phase, the toxin is released, and it's an exotoxin. So this disease, diphtheria, is prevented by this vaccine, the DTaP vaccine, diphtheria toxoid and pertussis. And this vaccine, and it's tetanus as well. This is a vaccine against not the bacterium, but against the toxoid. So it teaches our immune system to recognize this exotoxin. And as long as the exotoxin is not operating, our immune system is able to overcome the corynebacterium cells themselves quite easily. It is this toxin that gives the cell its virulence. So it has a systemic impact. It produces a rash, this erythrogenic rash. It produces a high fever, and it produces a virulent sore throat, an intense pharyngitis, and which is extremely painful. And it then causes the pharynx to produce this pseudomembrane here. Now look carefully. Here's the entrance to the airway down behind the uvula here. And this pseudomembrane could become so thick that it could actually occlude the airway, especially if it was on both sides, obviously. And that is how children died. They suffocated in a terrible, slow, agonizing suffocation as the pseudomembrane was produced. There were doctors who specialized in removing the pseudomembrane, which was an agonizingly painful operation, but and was not always successful, because by that time often children were so weakened that the systemic illness went on to kill them. A very, very bad disease in children indeed. So this has almost entirely been eliminated, at least in developed countries, by this diphtheria toxoid, tetanus, and pertussis vaccine, the DTaP vaccine. And it's usually given in childhood, obviously, but we may have boosters throughout our lives. Whenever you go for a tetanus booster, very often that tetanus booster will include a diphtheria toxoid as well, so that it boosts your both immune responses. In the United States, around about the turn of the century, there were less than 5 cases of diphtheria recorded per annum in the United States. Recently, however, that number has begun to increase because this vaccine is one which has received particular attention from the anti-vax community. And many parents now are refusing to have their children vaccinated. And I would venture to say they don't actually understand what the consequences of that lack of vaccination might be. Okay, so there's just one other one I'm going to mention now. This is just a reminder of your anatomy. This one, we're going to talk about what the impact of infections in the middle ear here. And the middle ear is connected to the pharynx by this tube called the eustachian tube, sometimes called the auditory tube. It is normally patent, and it allows not only drainage from the middle ear, but it allows us to equalize the pressure between atmospheric and this almost enclosed interior chamber, the middle ear, which holds the bones which communicate to the inner ear from the eardrum. So if this becomes closed, then it can lead to the situation where bacteria reproducing inside the middle ear begin to accumulate. They cause inflammation, they cause the release of fluids into the middle ear and the accumulation of pus. And that begins to increase in volume, and it causes a swelling of the tympanum and produces this agonizing earache. Very, very common in children because their eustachian tube is much narrower than ours, than in an adult. So it's much easier to block it. Even a mild sore throat can, in fact, produce enough inflammation that it will close off the eustachian tube. So this causes this condition, otitis media. Otitis media is inflammation of the middle ear, and it can be caused by a large number of different bacteria. But commonly, it is produced by Streptococcus pneumoniae, which is again maybe normally part of the microflora, despite its terrifying name. But also other ones, Streptococcus pyogenes is definitely also implicated. Another one here called Haemophilus influenzae, which I'll have a word about later. It actually has nothing to do with influenza, don't be confused. This can also be produced by the respiratory syncytial virus that we talked about before. And this is one of the complications when children have RSV. So it's treated with broad spectrum penicillins and that sort of thing. I'll show you here. This here is the tympanum here. And it's a bit difficult to see because we can't see it in 3 dimensions. But this is like a balloon facing out into the external canal of the ear. And as nurses, this is something you're going to often see. Very, very, very typical and very diagnostic of otitis media. And it's caused by this intense pressure inside the blocked middle ear. If this does not respond to antibiotics and anti-inflammatories, that sort of thing, then what they will often do is they'll put in little plastic grommets, which create an artificial hole in the eardrum. And it relieves the pressure and allows for drainage until the inflammation heals. And those grommets don't have to be removed. They will just be shed in the normal course of the maintenance of the eardrum. So that is a common solution, to insert grommets into the eardrum. Okay, so now we're going to leave the upper respiratory tract, go on to the lower respiratory tract. And here we begin to talk about things which are extremely serious. Any infection of the lower respiratory tract has the potential to become extremely serious, if not life-threatening. So Inflammation of the bronchi is bronchitis, and that is familiar to most of us. And inflammation of the bronchi can be bacterial, but is very often viral as well. And it produces the effect that, in fact, we start coughing vigorously. And often that coughing can exacerbate the situation because it begins to irritate the mucous linings of the bronchi and causes further inflammation. Bronchiolitis is rather more serious, and that is inflammation of the smaller bronchioles. And the reason that it is more serious is because inflammation of these small bronchioles can actually isolate the alveoli and cause blockage of the alveoli. So the drainage from the alveoli fails, and any bacteria inside there, which happen to be trapped inside there, reproduce, begin to cause inflammation in the alveoli themselves, cause accumulation of fluid in the alveoli, and that is the classic situation of pneumonia. And consolidation of the alveoli means that the alveoli can no longer perform the function of sending oxygen to the body and voiding carbon dioxide. So that is a very, very serious situation. There are various things that can cause this. The first one to talk about is another of the childhood diseases, which used to be extremely common, and this is pertussis, or whooping cough. And it's caused by this organism, Bordetella pertussis. Bordetella pertussis is gram-negative rod, and it is another of these bacteria, which in and of itself is not particularly pathogenic unless it has these virulence factors associated with it. And Bordetella pertussis actually produces several different toxins, and some of these are cytotoxic. It has a particular affinity for the cilia in the bronchi, to which it attaches. Here they are here. These are the cilia, and the red cells here is Bordetella. They're attached to the cilia, and they produce a tracheal cytotoxin. And this has various, if these cytotoxins have various effects. First of all, they begin to kill off the cilia. They stop their motion, and they shut down the ciliary escalator. And the other toxin enters the bloodstream and actually causes systemic effects. So it is a multi-effect that results from pertussis. The inflammation of the bronchi, but the failure of the bronchociliary escalator, the mucociliary escalator, means that the mucus begins to accumulate inside the lungs, and it induces intense coughing. And the child will get into paroxysmal coughing. And paroxysmal coughing, here it is here, the paroxysmal stage, they will cough to such an extent that they will void all the air inside the lungs. They often curl up in a ball and then sit back to try and draw a breath, and they'll pull this sound. And that is where the name comes from, the whooping cough. And that is a sign that they are struggling actually to gain air into the lungs. The convalescent stage, if a child has suffered from severe pertussis, they may be ill for months. And that is largely due to circulating toxins. So this is another of the diseases which is prevented by this vaccine, DTaP, and it's the P part, pertussis, but it can be treated with antibiotics as well. Prevented by the vaccine, but it can be treated by antibiotic. So again, because this is the target of anti-vax groups, the pertussis had become very uncommon in the developing world and is now in pockets making a reappearance. Okay, so now we leave most of what we're talking about actually have been the kind of childhood diseases. Very, very important because they can have tremendous impact on childhood mortality. But now let's have a look at some of the more general lower respiratory tract infections. And the first one we're going to talk about is probably one of the most important diseases in the human population that has ever existed. And we know that it has existed both within humans, Homo sapiens, but also in some of our associated evolutionary cousins like Neanderthals. There is signs that they too suffered from tuberculosis. In human beings, it is still a major cause of human morbidity and mortality worldwide. We did have a golden era in the 20th century when we believed that we were on our way to eliminating tuberculosis as a major source. And that was because of the generation of very effective antibiotic treatment. But unfortunately, tuberculosis has the ability to rapidly develop resistance to antibiotics. And this has been a continual battle. There's one other thing which I'll mention later on, which explains its reigniting in the human population as a major threat. So it's caused by this organism, Mycobacterium tuberculosis. And you'll remember that the mycobacteria are those organisms. They are fundamentally gram-positive, but you can't gram-stain them because they have a wax called mycolic acid in the cell wall. And they have to be stained through a special technique called the acid-fast stain. And these are obligate aerobes. Now, this is going to become important, as you'll hear in a while. They have a very slow growth rate. About the generation time is about 20 hours. Now, this is important not only in terms of disease progression, but it's also important in terms of our ability to actually diagnose. And they're very difficult to grow, and they grow extremely slowly. Those mycolic acids, the lipids in the cell wall, are not there just by chance, by the way. They are actually confer several properties on the bacterium. For example, they are very resistant to drying and can survive for a very long time on a surface, on a fomite. They are also resistant to many antimicrobials. And in particular, they are resistant to the antimicrobials which are produced by our own immune system. This bacterium has existed together with human beings or our relatives for a very, very, very long time, for millions of years. And they have developed a mutual battle plan. Our body, our immune system continually tries to recognize mycobacterium and deal with it. Mycobacterium has its very sophisticated responses to our own immune response, which dampens it and which allows tuberculosis often to evade our immune response. There are some other mycobacteria which can also produce similar kinds of disease in human beings. The first is Mycobacterium bovis, and that is bovine or cattle tuberculosis. And that can actually be acquired as a zoonosis by human beings. But it's relatively uncommon. It used to be much more common in the 19th century and before. But nowadays it's less than 1% of the US cases. But it is interesting to note that Mycobacterium bovis was very common in Europe because it is easily transmitted by milk as well as by meat products, but especially by milk. And this was one of the reasons why Pasteur, the great French microbiologist, developed the system of pasteurization, of heating up fluids to a point where you killed off most vegetative bacterial cells. And in doing so, Mycobacterium bovis was eliminated from the milk supply. And that was one of his great achievements. There is another one called Mycobacterium avium intracellulare, and very commonly just abbreviated as MAI. And MAI it never occurs in human beings. It just doesn't. It does not occur in human beings unless they suffer complete immune system collapse. Normally, if we're exposed to MAI, which we probably are all the time, our immune system immediately recognizes it and kills it off. But in people whose immune system has collapsed, like late-stage HIV infection, it will produce a pneumonia, a lung disease, which often proved fatal. And it was MAI was one of the marker infections in the early days of the AIDS epidemic. It was one of the markers which told clinicians that this person had progressed to the late stages of HIV infection. Okay, so here is the bacterium. This is a stain from a sputum sample. They get people to cough from as deep in the lung as possible and to try and produce mucus from deep in the lung. And that is then stained with the acid fast stain. Now go back, please, and look up what that acid fast procedure is. I think you did it in lab. It consists of heating the sample, the slide, with carbofuchsin. And when you heat carbofuchsin, it melts the mycolic acids and it can penetrate into the cell. You then cool it so that wax is set again and they seal the carbofuchsin into the cell. You then wash with acid alcohol, which very easily washes carbofuchsin out of all other cells on the slide, and you counter stain with the methylene blue. And what you end up with, these red cells are said to be acid fast. And the blue, all of this blue, these are all, this is all epithelial cells and mucal proteins and things like that in the background, all stained blue. And they have this characteristic pattern sticking together, stacking themselves together like that to form what's called corded growth or palisade. Okay, so tuberculosis is a really terrible disease. Surprisingly enough, it is not particularly contagious, but it is commonly associated with situations where people are living in close quarters with one another, where if one person in the group has pulmonary tuberculosis and just in ordinary course of coughing, speaking, whatever, producing droplets with the bacterium in it, they can be inhaled by other people, and an infection can result. But it's important to say that an infection does not necessarily result in a serious illness for reasons that I'll show you now. The first thing that happens when Mycobacterium is inhaled deep into the lung is that it is very easily recognized. The immune system is trained, in effect, to recognize the presence of Mycobacterium, and the first thing that will happen is it will get eaten by a macrophage. Now, and in normal circumstances, any bacterium which gets eaten by a macrophage is immediately digested and is used as a food source, but for other things as well. But in case of tuberculosis, the cell can actually evade that digestion process and can actually begin to reproduce inside the macrophage. The macrophage, in actual fact, will respond to this presence, the continued presence of the bacterium, by summoning many other macrophages. and they begin to cluster around the infected cell. And they form this, this cluster is called a tubercle. And that's where the disease gets its name. And tuberculosis is the proliferation of these tubercles. And the tubercle is attempting to isolate the bacterium in the middle, isolate it from the rest of the body. In some cases, this is entirely successful. And this tubercle then becomes calcified and isolates the bacterium completely, and it never proceeds any further. And that is the case for many people. They have these tubercles on their lungs, but there is no chance of them getting diseases. The bacteria can last in a sort of dormant state for a number of years, but eventually the whole thing just becomes completely calcified. However, the other course of pathogenesis is that the macrophage, the initial macrophages actually die and release cells. And they release cells, the mycobacterium cells, into a sort of a cavity that begins to form inside the tubercle. And the bacteria then begin reproducing, and they form a liquid center in the middle. And if it gets arrested at this stage, all is well and good. It can just stay there. But very often, the tubercle now begins to break through into bronchi that are adjacent to the tissues. So whether it stays as this latent kind of tubercle that gets calcified, or whether it proceeds to active disease, is the source of much research, because it's not actually clear what the triggers are that allow one or the other course. But once the bacteria are released in the bronchi, they also begin to spread to other areas of the lung, and they now begin to cause the actual decay of lung tissue. They begin to form cavities in the lung. They kill off the lung tissue itself. And part of that is because of virulence factors of the bacteria. Part of it is actually because of immune responses to their presence. These cavities and spots of infection, as the bacteria spread into the other bronchioles and bronchi, they begin to spread throughout the lung until there is a disseminated tuberculosis situation. Often it will remain isolated to one lobe for quite a long time. And one of the approaches used to be in the old days to collapse that part of the lung, or to collapse the whole lung and hope that you consolidated the infection and prevented its spread to the other lung. So I just want to tell you that this is a very virulent disease. Even though it reproduces slowly, it causes massive damage to tissue. It can spread then to the rest of the body. It can appear in virtually any tissue, in which case it's called miliary tuberculosis. There have been numerous attempts to develop a vaccine, and the only moderately successful one is this one called Bacillus, the BCG vaccine, for Bacillus calmette-guerin. And this is attenuated bacterium. It is attenuated Mycobacterium bovis. That means they grew it over and over and over and over again on media, not in a living organism. They grew it over and over and over again until it lost its virulence factors. And they then developed that as a live vaccine. But unfortunately, it has some effect in children, but the effect does not last. And in adults, it is very ineffective. And in the United States, it's not administered at all. So there are various tests to see whether you have tuberculosis or have been exposed to tuberculosis. One of them is this one. This is a common one. This is the tuberculin skin test. And all that they do is they just inject a small amount of tuberculosis bacteria protein, not the bacteria itself, but proteins from the cells. And then they watch to see if you've been exposed and you have antibody, then you develop a welt right where the injection site is. But this is one of the reasons why the BCG vaccine, we talked about before, the Bacillus calmette-guérin vaccine, is not administered in many countries, because if you've had the BCG vaccine, you can't have the tuberculin skin test. Because if you've got to have the BCG vaccine, you will have the antibodies, even if the antibodies are not particularly effective. Then the classic old tests are the sputum tests that we saw before, and that they would simply examine for acid-fast bacteria, and coupled with culture of the sputum, which is very difficult to do, a very long-term process. But nowadays, blood tests and serology tests are available in order to check for bacterial DNA and antibody response and degree of antibody response, which gives you an indication of previous exposure, but also of active exposure. Treatment of tuberculosis can be extremely difficult. It always entails a very, very long-term antibiotic treatment and usually a minimum of 6 months of drug therapy. And the old drugs that were used were these: isoniazid, rifampin, ethambutol, pyrazinamide. But I don't need you to know these names. Okay, just know that there was a group of antibiotics which were used early on for some years, a decade or more, which were extremely effective, but then began to lose their efficacy. And within populations, it began to see disease which was entirely resistant to these antibiotics. So they moved then to second-line drugs, and these are them. Streptomycin might be the only one that you've heard of, but these second-line drugs were also prone to the development of resistance, unfortunately. So when the appearance of the initial resistance were called multi-drug resistant strains, because they serially became resistant to each of these as they were used. And so those were multi-drug resistant. But now we have extensively drug-resistant strains, which are resistant to first-line and second-line drugs. And these are the subject of much research, but also the source of an enormous amount of new disease. The incidence of tuberculosis in many parts of the world is increasing, including in the United States. So here, have a look here. It is a major human disease and is a source of major human mortality. Over 2 million people die of tuberculosis a year worldwide, and 10 million, the incidence is 10 million per year. So that is the incidence. The prevalence is obviously much higher. And 1/3, this is an extraordinary statistic, but 1/3 of the world's population have latent TB. That is, they may not have active disease, but if you look at their lungs on X-ray, you will see tubercles there, and tubercles which have not become completely calcified have the potential to form disease. Should they ever, what is happening in those tubercles is the immune system and the bacteria have established a balance. The immune system is suppressing the spread of disease and containing the bacteria. If that balance should become interfered with, and the structure of the tubercle and its maintenance breaks down, those bacteria can be released to go on to form fulminant disease. So here is the real problem. 1/3 of the world's population has latent TB. And that latency is maintained by the immune system. This is how TB and HIV become intimately linked. So what I want you to look at is the prevalence of TB in, for example, in Africa, in India, and then throughout Indonesia, all the Far East, etc. Look particularly at Africa, because Africa is a case study of the involvement of HIV and tuberculosis. If people already have latent TB, and they then acquire HIV and their immune system collapses, the structure of those tubercles breaks down and they develop fulminant disease. They then become infectious and they begin to spread the disease. This is one of the reasons, if I was to take out the sign saying tuberculosis for this graph and replace it with the sign saying HIV, for Africa, it would be pretty much the same. They pretty much plot together the incidence of AIDS and the incidence of tuberculosis, because they are so closely linked in pathogenesis. It's a very, very dangerous and a very tragic situation. In Africa, we had just begun to feel in the 20th century, mid-20th century, that the prevalence of TB had decreased so much that we were on the way to eliminating it, or at least dealing with it successfully. But that has completely been lost with the advent of AIDS through the population. There has been an explosion in the number of tuberculosis cases, and especially in the number of extensively drug-resistant tuberculosis. So the two things in Africa have combined to produce an absolutely tragic epidemic of tuberculosis associated with HIV. Okay, so let's just have a look now at some of the other the lower respiratory tract infections. And of course, one of the most important are the bacterial pneumonias. Now, pneumonia is really a sign. It's not a disease in and of itself, it's a sign. And it's a sign that the alveoli have become flooded with fluid, inflamed, and are no longer able to perform the normal task of exchange of oxygen and carbon dioxide. One of the major causes of pneumonia, in fact, so common that it is called typical pneumonia, is another streptococcus. And this is Streptococcus pneumoniae, again, a common member of our normal microflora. And there are other causes of pneumonia, which are called the atypical pneumonias. And so one really important one is healthcare-associated pneumonias, and in particular caused by Staph aureus, which is a different kind of pathogenesis. So when the pneumonia only, the lung is divided into lobes. When it only infects a particular lung, that is called lobar pneumonia. But it can actually infect several of the lobes of the lung. Bronchopneumonia is infection of the lung, which begins to involve inflammation of the main bronchi as well, and that is called bronchopneumonia. There is an associated condition called pleurisy, and this is an inflammation of the pleural membranes which surround the lungs, the double layer pleural membranes. They are responsible not only for protection of the lung, but for creating a slight negative pressure within the lung to keep the alveoli inflated. And the pleura can become inflamed rather, as a result of an immune response to inflammation in the lung. And this creates an intensely painful inflammation of the pleura, which can actually exacerbate the pneumonia by making breathing really difficult. So that is its main impact. So the most common organism to cause these is Streptococcus pneumoniae, and these are often referred to as the pneumococcal pneumonia, And this is a common effect of long illness and long bed residence with insufficient lung hygiene, respiratory hygiene, which allows for accumulation of fluids, etc., within the alveoli. Streptococcus pneumoniae has often been called the old person's friend because it was rather cynical, but it refers to the fact that very often this is the final illness which very elderly people suffer because they become bedridden. It becomes very difficult to manage lung hygiene. And it's the thing that finally carries them off. It's gram positive. You should remember that. We've talked about this before. We actually talked about this when we talked about DNA, because workers looked at trying to develop a pneumococcal vaccine and recognized that there were two strains of Streptococcus pneumoniae. There was one strain that had capsule, which is pathogenic, and one which did not, which was non-pathogenic. So it's the pathogenic one we're really interested in, and that has a thick capsule. You are here. This is the capsule here. Here is the cell. The deep blue here is the cell, and around it is the capsule. The capsule enables the cell to stick to the mucous surface, but it also allows it to evade the attention of the macrophages. So if it's conducted deep into the lungs, into the alveoli, it can begin to cause an inflammation, causing the alveoli to fill with fluid, red blood corpuscles, etc., to consolidate and lose their function entirely. It's diagnosed by just standard clinical mechanisms like X-ray, also auscultation. You can actually hear what's going on in the lung, and you can also detect its presence by looking for capsular antigen in the urine, which is a quite sensitive test. It can be treated with antibiotics and also with physiotherapy, of course, to try and improve lung hygiene, to clear the lungs and get fluid expressed. But the most important thing about it is that there are good vaccines against it. And there are two different pneumococcal vaccines, which are extremely effective, which everybody should have. Anybody can get pneumococcal pneumonia. These pneumonias can be caused by other organisms as well. So this one, I said that I would refer again to this. This is Haemophilus influenzae. Influenzae, it has nothing to do with influenza, except that during the great 1918 influenza epidemic, they were desperately searching for a cause of the virus. Was very poorly understood at that time. And one of the workers on the epidemic discovered this Haemophilus influenzae, this Haemophilus in numerous patients who had the flu, and assumed that it was this that was causing it. Wasn't. It was a secondary infection after the influenza epidemic, after the influenza infection. There is a vaccine against it. It can cause a very serious pharyngitis as well, bronchitis, and it can then spread to cause a pneumonia. And that is extremely important in children. And Haemophilus influenzae will often spread through a population of infants or very young children. There is a vaccine against it, and it has now become relatively uncommon, although not everybody knows about this vaccine. So it still definitely does occur. And it can be treated with antibiotics as well, quite strenuous antibiotics. The last pneumonia that I'll mention is this one. This is mycoplasmal pneumonia. Actually, this is not the last one. I'm getting confused. Mycoplasmal pneumonia. Mycoplasma pneumonia is caused by Mycoplasma pneumoniae. It's a very, very, very strange organism. It's one of the few bacteria which has no cell wall. And you'll remember that the cell wall is the bacterial cell's major osmotic control. So Mycoplasma pneumonia, very small organism, and but it is isolated to situations where it can be in an isotonic situation, and it causes a mild pneumonia. It's often called walking pneumonia because people have low-grade kind of grumbling kind of illness and cough and everything, and it's only a doctor will recognize it by listening to the lungs and saying, oh, you've got pneumonia. And it's usually self-limiting. It has a very distinctive look when you can culture it, and it forms these what they call fried egg colonies. They look just like a fried egg in a frying pan. So it's if it needs treatment, it usually in a healthy young person, which is very common. It often spreads through residence dorms and army barracks and things like this. It can be treated with tetracyclines, but usually no treatment is required. It's just of interest. And here's the last of the pneumonias, but this one I have talked about before. And this is the disease caused by Legionella pneumophila, but it can also be caused by other species of Legionella. And Legionella is an aerobic gram-negative rod. Grows very well in water, air conditioning, cooling towers, humidifiers, all sorts of things like that, and even in water fountains. And is part of a community of organisms that form biofilms within these mechanisms. But if it gets caught up in the water, in the fluids in the systems, it can be spread and it can actually cause an infection. And so people are infected by inhaling aerosols. It's not transmitted. It's not contagious at all. And under normal situations, in a young person, it is also usually self-limiting, but it can cause a very serious, highly inflammatory pneumonia, and is of particular danger in the elderly. And unfortunately, Legionnaire's disease is commonly associated, or it used to be commonly associated with any building that had central air conditioning, because the bacterium could be conducted throughout the air conditioning system. Nowadays, modern air conditioners are designed so that they minimize the likelihood of the growth of Legionella. So we're going to talk now just briefly about this organism, chlamydia. Now, chlamydia is a very interesting organism because it's one of the very smallest of all of the bacteria. It is about 100 times smaller than an E. coli cell. It is so small that it actually contains almost the bare minimum number of genes in its genome and the minimum amount of cytoplasm and cellular machinery for a cell to survive. And it is an obligate parasite. That is, it has to gain entrance into a host cell and utilize the host cell's machinery in order to truly survive and reproduce. What does that sound like? It sounds very like a virus, excepting that chlamydia has a DNA genome, it has a metabolism, and it has cellular mesh, some of its own, it has its own ribosomes, for example, things like that. But it is very interesting to us because it may be from something like chlamydia that the virus is developed or evolved. That's just of interest to you. But I'll show you. Here is a host cell which has been infected with chlamydia and it's reached maturity. The cell lyses and it releases these very small they're called elementary bodies, which attach to the cell membrane of the host cell and induce phagocytosis, just like the viruses. Here it is here. But they actually survive inside this vesicle and begin to reproduce. And they undergo binary fission, form a huge number of what are called reticulate bodies. This is about as big as the chlamydial cell is ever going to get. The reticulate bodies then begin to divide into this very, very small elementary bodies and eventually cause the cell to lyse. Now, there are several different kinds of chlamydia. And one that I really want to mention to you because these are so popular, one of them is associated particularly with parrots, and this is Chlamydia psittaci. It's a gram-negative. As I've told you, it's obligate intracellular bacterium. It's an obligate parasite. And that can be transmitted by these elementary bodies from the bird to humans. And it starts with fever, headache, chills, those sort of things. But it can produce an extremely serious pneumonia, an extremely, extremely serious. This can be a very serious disease. So one needs to be a little bit aware. If you have parrots or whatever, you need to be aware that this can occur. It's treatable with antibiotics, fortunately. Interestingly enough, if we want to culture chlamydia, we have to do it in eggs, in fertilized eggs, just the way that viruses are cultured. There is another one called chlamydia pneumoniae, which is actually a contagious chlamydia, and but it causes only a mild disease, and it's usually in young people. It looks very like walking pneumonia. The only thing about it is that there is some indication that it predisposes people to atherosclerosis, that is, to inflammation of the blood vessels and to development of plaque and clotting along the sides of blood vessels. And the last one I'm going to mention is one which in a way sits with Mycobacterium avium intracellulare, in that this is a pneumonia which is extremely uncommon under normal circumstances in human beings. And this is pneumocystis pneumonia, or PCP. It is caused by this organism, Pneumocystis jirovecii. And this is a strange thing. It is actually I only mention it, it's not a bacterium. I only mention it because it is so strongly associated with the disease of AIDS. It is a diagnostic organism for AIDS, and it only occurs in human beings where the immune system has collapsed. And we're probably exposed to it the whole time, but it never develops, it never breeds and never reproduces because our immune system knocks it out. But it actually can cause this very, very serious consolidation of lung tissue and was a frequent cause of mortality in those who had HIV. That is just out of interest to link it together with MAI as being these kind of indicator organisms in people whose immune systems have collapsed. Okay, and that's it. And so I will see you next week, and I wish you all a very, very happy Thanksgiving.